Ina mana, ina reo, ina iwi me na hoe wha, no mai harimai, tēnā koutou katoa. Ko Helen Nicholson, tōku ingoa, te tumu whakarai, taupua, te whare wanaka o tākou. Nō reira, tēnā koutou, tēnā koutou, tēnā koutou katoa. Kia ora, my name's Helen Nicholson and I'm the Acting Vice-Chancellor of the University of Otago. It's my great pleasure to welcome you all to this special occasion to celebrate Ping Lu's promotion to Professor. And it's good to see so many of Ping's colleagues, students and friends, but I'd like to give a special welcome to Ping's family, her parents and brothers in China, Ni Hao, and to her family here with us. So her husband, Hu, her sons, Wei and Xing, her daughter-in-law, Jesslyn, and her parents, Eka and Mimi, her niece, Tong, and her brother and sister-in-law, Ming and Iwen. It's lovely that you can join us all. So some of you will have heard me say before that promotion to Professor at the university is not easily achieved, and Ping has gone through a rigorous process to demonstrate that she is worthy of this promotion, a process that involves seeking feedback from external referees across the world. And inaugural professorial lectures are always wonderful opportunities for us all to take time to learn about the research our amazing colleagues do. This IPL is particularly special to me because I was head of a department of anatomy and on the panel that appointed Ping as a lecturer in 2005. And as a consequence, I've followed her career and development with interest. So while Professor Hyland's going to provide a formal introduction, I'd like to add a few personal comments about Ping first. So Professor Lu is a gifted neuroscientist, and she and I used to have labs very close to each other. And I quickly saw her strong work ethic and the care that she gave to her students. And these qualities have resulted in many publications and also international collaborations. In terms of her teaching, when Ping first came to anatomy, she already had a strong research track record, but had very limited teaching experience. And I'm sure she won't mind me telling you that her early teaching evaluations were not stellar. However, it's her response that impressed me. She sought advice and help, put in an amazing amount of work, and now her teaching evaluations are some of the best. And as one of her referees comments, she's an outstanding educator. As well as being a busy academic and a mum, Ping has also made significant contributions to the community in Dunedin, particularly the Chinese community. So when her children were younger, she was involved and at one stage the president of the Chinese language school. She's a member of the Chinese Cultural and Arts Association, and you can often see her performing at various Chinese festivals. But she's also contributed to the life of the university. So she's part of the Dunedin Chinese Student and Scholar Association, and she and Hu have quietly supported many international Chinese students, as well as assisting the university with hosting our visiting delegations. So thank you, Ping, for your support. So it's been a pleasure for me working with you. I'd like to add my congratulations to your promotion, and I'd also like to ask Brian to come and formally introduce you.
Tēnā koutou, tēnā koutou, tēnā koutou katoa. Greetings to you all. It is indeed my great privilege and pleasure to have the role to introduce Professor Ping Lu's inaugural professorial lecture. Ping graduated with Bachelor of Medicine in China in 1985 and worked as a physician for 8 years in clinical geriatric practice before completely removing herself from China, migrating to New Zealand and embarking on a complete transmogrification change, it's too complicated a word, in Korea. So she embarked on a completely new direction in neurobiology research, embarking on a PhD with Professor Dave Bierke here in the Department of Psychology. She completed that in good time in 1997, and in fact, it was an outstanding PhD. And on the back of this outstanding PhD, in which she published 7 full papers, she received a 2-year Health Research Council Career Development Postdoctoral Fellowship in 1999, which she used to support further work in Professor Bierke's laboratory, further postdoctoral study. That was followed by 5 years postdoctoral research in the Department of Pharmacology and Toxicology over with us in the School of Biomedical Sciences, in the laboratories of Professors Paul Smith and Cynthia Darlington. And as already noted, following that, she was sort of snapped up by the Department of Anatomy and appointed to a lectureship in 2005, promoted to senior lecturer in 2010, and associate professor in 2015. And as we've noted already tonight and on multiple other occasions, promotion to professor requires sustained outstanding leadership and performance, as assessed not only by our local committee, promotions committee, but by international independent reviewers. So just quickly summarising some of the highlights of Ping's achievements. In teaching leadership, which has been mentioned already, at divisional level, Ping has served a long-standing role as convener for the nervous system block module in the MBCHB, the medical program, which is not part of the school, but is a contribution that she makes into the medical program as part of the Division of Health Sciences. And that's a major challenge, not only because it's considered by the students the most challenging content-wise, but also it involves a whole lot of clinicians and academics who have to be marshalled into the appropriate place at the appropriate time, year on, year out. So it is a major contribution to medical training here in New Zealand that Ping has provided over those years. Within the department, she served as chair of the Departmental Teaching Committee and convened multiple papers over the course of her career in the department. And as we often say, we combine into teaching, that is nurturing our postgraduate students through into their careers. And Ping has an excellent record there as well, having mentored multiple PhDs to completion. I think she's got 5 on the go at the moment, 3 master's students, multiple honours students in her laboratory, and they've displayed very high levels of achievement, reflecting the level of support that Ping provides to them. Now, in research outputs and recognition, Ping has funded her work with a succession of external grants, including being co-principal investigator on an HRC program grant, and project funding from the Neurological Foundation of New Zealand and MBIE. And she's engaged, as all of our professors do, but it's always worth commenting on, local and international collaborations, which mean that she's feeding into others' research and taking from others in terms of building the research profile here in New Zealand. So all this input, money, collaborations and so on, has led to a string of high-impact publications, over 70 peer-reviewed journal articles, with a very high proportion of them as first or senior author, reflecting leadership of the research. Highly cited papers. So H-index, for those of you who are interested in those sorts of numerics, of 29 at the time of applying for promotion. And these are occurring in journals such as Neuropharmacology, Translational Psychiatry, Neurobiology of Aging, Oxidative Medicine, Schizophrenia Research and Scientific Reports and so on. And those titles reflect not only the quality of the journals, but also the subject matter of her research and its link through to clinical application. So Ping's research has focused on the role of arginine metabolism in the aging of the brain, in normal aging, but how altered metabolism of arginine may contribute to degenerative disorders, such as Alzheimer's and other chronic problems, including neuropsychiatric disorders. And as Ping will no doubt explain, arginine is an amino acid, structural component of proteins in the body, and it's associated through its metabolic pathways with the generation of many other bioactive molecules, such as nitric oxide, agmatine, and polyamines. And a particular emphasis in Ping's work is exploring, on the one hand, how the levels of those substances in the body and how changes in their level could be used as biomarkers, in other words, ways of tracking the progression or the prognosis of a disease that could be accessed easily from the body fluids, but also, on the other hand, how some aspects might be protective of disease and therefore could offer potential therapeutic approaches. For these studies, she brings to bear a wide range of methods, extending from animal models to work in human samples and post-mortem tissue. So in her lecture this evening, Ping, I'm sure, will give us an overview of her research, but she wishes to focus in particular on her work on arginine and its metabolic pathways in processes of normal aging and in neurodegeneration and the potential clinical applications. Once again, it's my pleasure and privilege to introduce to you Professor Ping Lu to deliver her inaugural professorial lecture. Tēnā koe.
Tēnā koutou, tēnā koutou, tēnā koutou katoa. Thank you very much, Helen and Brian, for your very kind introduction and also all of your support over the years. So life is a journey. My journey has not been simple and easy, I have to say. I would like to use this opportunity to thank you all for coming, and I really want to thank everyone who has supported me on this journey. So thank you very much for coming and for this very special occasion of my academic life. So today is a very special day. It's the 20th of May. In Chinese, we call it 520, which means, 我爱你, I love you. So I just want to say I love you all today. Okay, and especially I want to thank my families. I love you all, my 3 boys, my husband Hu, and my 2 boys Wei and Shane, and my lovely daughter-in-law Jesslyn. So I used this photo, it's her wedding photos. And also her parents, Mimi and Eka, they are here today. Thank you very much. Some of you probably don't know, I have quite big family in New Zealand. My brother-in-law, Associate Professor Ming Zhang, and my sister-in-law, Ewan, and their family, Chu, Ollie, and Max, and Han. My sister-in-law, May, and her daughter, E, and this is my younger brother, and his daughter, Tong. Tong has been here with me for 5 years, so I think, Tong, you are my daughter now. Okay. I want to thank to my family in China. So this is my father and my younger brother, Liu Jian, and his wife, Yong Mei, and another younger brother, Liu Yong, and his wife, Jian Lan. They are going to watch my IPL live stream. I don't think they understand Chinese, but I do believe they must be very, very happy to see me on the screen. Okay, so I want to thank my mother, Zhou Lanying, who passed away many, many years ago. And I want to thank my parents-in-law, Zhang Li and Zhou Ming. They have supported me enormously during my journey. So you have left us, but I don't think you really have left us, and you live in my heart forever. So just in case if I run out of time, I would like to thank my team, my current and past student, everyone on the on the screen, and also staff members highlighted in the red box. Thank you very much for all of your hard work. Okay, I am here today is really because of you. Thank you very much. I also want to thank the Departments of Anatomy, Psychology, Pharmacology, and Toxicology at the School of Pharmacy. And thank founding bodies and a couple of research centers, Health Research Council, Neurological Foundation, Lottery Health, Brain Health Research in New Zealand, and Brain Health Research Center. And NCDCRCC is long-term. It's a New Zealand and China non-communicable disease cooperation center. Okay, for all of your support. So my early life, I was born in Anhui. So this is a photo, I think 100 day. I was at 100 day. As you can see from this map of China, my parents were separated 4,000 kilometers away. Okay. So when I was young, my mother basically looked after me and also my two younger brothers. So this is a photo when I was 3 years old with my parents. I couldn't find many family photos with my parents just because my father could only visit us once a year, about 1 month, but he had to spend about 8 days on the train. So my two younger brothers, they were 3 years and 5 years old younger than me. So most of the time, my mother had to look after us, which is very, very difficult. And sometimes my grandmother came to help. And sometimes my mother sent us to my grandmother's place. So it's a kind of a mixture travel around. So when I was 10 years old, finally my parents got together. So my whole family moved to the south part of Anhui and called Huangshan Yixian. And Huangshan, called Yellow Mountain, is the most beautiful mountain in China. It's very famous because it has the sea of cloud and very beautiful rocks and also pine trees. So my hometown is also pretty and it has been regarded as a paradise in the world and a painting in the countryside. It was a very beautiful place. And another thing is, my hometown has so many, probably over several hundred, well-preserved Qing and Ming Dynasty houses. Okay, it's a very, very special place. I was in Yixian for 6 years. I finished my high school. So this is my high school, my class. So this is the top class, okay? And not many girls in that class. I did really well for the national college entry examination. So finally, I went to Hefei, which is the capital city of Anhui, for my university life. So those photos are me, and this side, I was 16 years old when I started the medical school. And I have to say, medicine wasn't my choice at that time. Was absolutely my mother's choice. Because soon after my family moved to Yixian, and my mother's health condition just declined dramatically. So he basically struggled a lot with her own health. So she would like me to do medicine and to be a doctor, so which can help the family and in the future can help my own family. So although at the start, medicine wasn't my choice, and later on, I thoroughly enjoyed all of the medical curriculum. I think in my year, we had 300 medical students. So this is my small class, 30 students, okay, in my small class, and you could only see 6 girls in the class, not many girls. So this is me in the front gate of Anhui Medical College. So now Anhui Medical College has been renamed as Anhui Medical University. So this is how the university looks like. So Anhui Medical University has always been the top university, top medical university in my province. So Anhui Provincial Hospital, I had my final internship year in Anhui Provincial Hospital. Then I worked there for 8 years as a physician. So this is the photo. So now the hospital looks like, and this is the old one. So we call the Department of Geriatrics, at that time it's called 高干病房, which what this means the department served for senior officer. I was lucky enough to be selected to work in this department because they want good people, responsible people to work for the senior officers. So this entire building actually was for the Department of Geriatrics. So most of the rooms in a single room or 2 bedrooms. It was very, very good, high standard at that time. So during this time, my life changed a lot. I found my husband, and 1 year later, I had my first child, Shane, and it was lovely. But sadly is my mother passed away in 1991 after 7 years in and out of the hospital because of her liver problem. So I would like to thank all of my colleagues in Anhui Provincial Hospital, and there was a very difficult time. I had lots of support from my friends and colleagues in this hospital, and I also want to thank Hu for going through this very, very difficult time with me, okay, for many, many years. So in 1993, October, I had to follow my husband. So Wei and I would basically traveled from China, about 10,000 kilometers away, and finally to Dunedin. And 2 months later, my parents-in-law, Lee and Ming, were here, okay? So at that time, my brother-in-law, Ming, and Ewan were already here, and the Waterhouse. So for quite some time, I think several months, everyone squeezed in one house, in Ming's house, for several months. So at that time, I really struggled. I was pretty worried about what could I do, because my English was really, really poor. And and Ming said, well, you probably need to think about and talk to people and find a suitable project for you so you could do like research, redevelop your career. Then I did. I sent a couple of letters around, and David Bierke, at that time he was a lecturer in the Department of Psychology, and I think we met 2 days ago after Dorothy's IPL. I asked David, well, you were so brave to offer me a full-time job. Why? What made you to offer me a full-time job? Because I had no experience at all for research. That was really an interesting experience. And David said, well, I still remember your first meeting, and you were very, very quiet, probably struggled with your English, and your husband did most of the talking. Right, so I worked with David for a couple of months, kind of a volunteer work, and then David offered me a part-time job. Then David got 3 year, I think it's HRC grant, and he was really, really interested in perirhinal cortex and spatial learning and memory. So I took the full-time job, and at the same time, I did full-time PhD with David. Good thing is, I did not let David down. I had 7 publications from my PhD, okay? And so I think I submitted my PhD thesis in January 1998, and David asked me to submit a hardbound one. And luckily, my thesis was accepted, no revision. So I was very pleased for that. So at that time, my big family had 3 people doing PhD, me, my husband, and also my sister-in-law, Ewan. As you can see, my parents-in-law, so they were here to help us to look after children, okay? I really want to thank them once more to help us to go through this journey. So why perirhinal cortex and spatial memory? So this is the human brain, okay? And this is the lateral view of the human brain. So the temporal lobe is here, and you can see this part and called a hippocampus. So if we make a cut, and you can see, this is the medial part of the temporal lobe, and there are a number of important structures for learning and memory. So the hippocampus, entorhinal cortex, perirhinal cortex, and also perihippocampal cortex. At that time, we know the hippocampus is really critical for spatial learning and memory. However, the role of perirhinal cortex in spatial learning and memory wasn't very, very clear. So my PhD used rat as a model. The rat brain is quite small. However, the general neural connectivity was very, very similar to human brain. So I carried out an electrophysiological study. So basically, I showed there is a direct projection from the perirhinal cortex to the hippocampus. And this pathway contributes to the lateral peripheral path, which is a major pathway from the cortex to the hippocampus. This pathway and also supports long-term potentiation, which is a mechanism underlying learning and memory. Then I did another set of study. So I basically used electronic, otherwise it's a drug lesion, okay, arbutinolic acid lesion, and make lesions specific to this area called the perirhinal cortex. Then test animals in the different kind of learning and memory tasks. So my work showed the perirhinal lesion produced spatial learning and memory deficits. So collectively, my work supports the perirhinal cortex contributions to spatial memory. I think this period was really, really helpful for me to have a really good understanding about the brain, anatomy, and also function, okay? And I learned a lot about the behavioral neuroscience. So after PhD, as Brian introduced, I had 2-year postdoc with David. I think I worked on the post-mortem cortex and pre- and paracerebellum. So those structures are the other structures that also belongs to the medial temporal lobe memory system. Then I applied for a 2-year fellowship, which is Health Research Council Career Development. And from now on, I worked with Paul and David. The project is about the perirhinal cortex and Alzheimer's disease. So this is my first project on Alzheimer's disease. So after that, the funding ran out. And one day, Cynthia came to me and said, we just received 3-year HRC grant. Would you like to join us to do something different? I said, yes. It was quite exciting. So I joined Paul and Cynthia's group and worked on the vestibular hippocampal interaction. So in our inner ear, there has a structure called, vestibular apparatus. So if there's any lesion to the vestibular apparatus can cause balance problem and can also affect the hippocampal function as well. So my work, along with my sister-in-law, Ewan, so she worked in Paul and Cynthia's lab as well. And we basically showed, some neurochemical changes in the hippocampus following this peripheral vestibular lesions. So at that time, Dr. Ian Appleton was in the Department of Pharmacology and Toxicology. He was the expert in the area of arginine metabolism. I think my work on arginine metabolism and brain aging was basically developed in that period. So I had my second son, Shing, at the end of 2003. So this is Shing, had your first birthday in the child care. So for my second child, the pregnancy was really, really difficult, okay? I almost died on the surgery table. I had a 5-liter blood transfusion, okay, when I had Shing. That was very, very difficult. But we are here, Shing. Right. So after PhD, I had 20 publications, and most of the papers, with myself as the first author. Although I worked very hard, I had a great time in David Bierke's lab and also Paul and Cynthia's lab. I don't know whether you can see, this is David, and you must be drunk. Can you realize you? So lots of parties, okay, and this is the Ewan. So if you don't know what vestibular appearance looks like, this is the vestibular, okay, the pink one. So that was a great time. So after that, I thought, well, I probably need to move on. I would like to make a change. I want to have my own lab. So I applied for the lectureship. So as Helen mentioned, my research record was great, but I didn't really have any teaching experience. Okay, basically zero. So when she was 14 months old, I joined anatomy. So thank you very much, Helen, for hiring me and also being a mentor for many, many years after that. So I'm going to make my boy a little bit embarrassed because Shane just got sick all the time. You probably don't believe we had to visit a doctor every month until he was 3 years old. So at that time, I basically prepared a very large yellow bag with everything inside. I'm really scared to receive phone call from childcare because if I receive phone call, must be something wrong with Shane. So that was very, very difficult because teaching was new. I had to set up a research lab and also look after this young child, right? But be strong. I am here now. Okay, pass through that very difficult period. So during my life in the Department of Anatomy, I have had a number of HODs, Professor David Green, Dorothy Allshort, she's here in the audience. We also have Peter Hurst, I think, as acting HOD for a short period, and Dave Gretton, Neil Gamble, and current HOD, Lisa, right? I really want to thank you all for your support over the years, and finally I have a lab. I also want to thank the Linda Ferguson fourth floor girls, Dorothy, Marilyn, and also Ruth Nepper. So during my early years in the department, I had lots of help and support from Dorothy, Marilyn, and Ruth Nepper. I want to thank Marilyn in particular because Marilyn at that time really was my shoulder. Her door was always open to me. When I had questions, I could just get in and Marilyn, help, she always helped. So thank you, Marilyn. So during my time in anatomy, I further developed my research in the area of arginine metabolism in brain aging and neurodegenerative and psychiatric disorders. So this is my research group, and we had some lovely photos over the years, okay, and some students came, go, and others still stay here. And thank you very much, as said before, and really your work, you have made me here today. Thank you very much. So now I'll talk about science a little bit. What is aging? Aging is an inevitable process, okay? So everyone has this process, born and grow up and be an adult. At some point, the functions of our body system basically declines. So my work is really just interested in this part of our life, and I am more interested in brain aging. So as you know, when we get old, we tend to learn slowly and probably forget quickly. This is the kind of age-related cognitive impairment. So rat or mice, they also showed similar symptoms. So therefore, we can use rat as a model to study brain aging. So this diagram just to show you the cognitive continuum. For some people, when we are getting old, we don't really notice any cognitive changes over time, okay? However, most people probably do experience certain kind of age-associated cognitive decline. That's all fine. This is normal aging. However, for some people, they probably show, more severe cognitive impairment, or it's called mild cognitive impairment, which is really relative to the people, in your age, right? They show probably more severe cognitive impairment. So most people can stay quite stable for a long time, and some people even have improved cognitive function over time. However, a small proportion, basically, their cognitive function just declined further, and we think this is Alzheimer's disease. So basically, MCI and also AD, we describe those conditions as pathological aging. So dementia is really like a big umbrella name, okay, to describe a group of cognitive impairment symptoms. So at the moment in New Zealand, probably there are 70,000 people suffering from dementia. It has been projected by 2050 there would be 1,070,000 people living with dementia. So Alzheimer's disease is the most common form, okay, under the umbrella of dementia. It is a neurodegenerative disorder characterized by increasing memory loss, and aging is a major risk factor. As you can see from this diagram here, at age 65, probably 1%, okay, of population in AD. However, you can see the numbers just increases when the age goes up. So if you compare the normal brain and also Alzheimer's brain, and you can see there's a significant atrophy, okay, relative to the normal. But if we slice the brain and use a certain method to look at some pathological profiles, and you can see amyloid plaques and also neurofibrillary tangles present in the brain as well. So sadly, Alzheimer's disease at this stage is no cure, and its cause is also not very clear at this stage. It has been proposed, amyloid beta probably plays a central and causative role in AD development. However, this hypothesis has been challenged increasingly over the years. So there are a number of challenges in AD field. For example, the exact cause of AD and early diagnosis, we don't really have a very good tool for early diagnosis, and prevention and also therapeutics. So I would like to give you a little bit background so you understand my following talk. I would like to introduce the first protein called amyloid precursor protein. So everyone in our brain, we have APP, okay? The short term is APP. And APP can be cut off by a couple of enzymes, just like physics. You can cut here or cut here. So normally, APP will be cleaved by alpha and gamma secretes to produce this very, very small peptide, which can be cleaved. So in the Alzheimer's condition, however, this protein is cleaved by beta different enzyme, okay, and also followed by the gamma secretes to produce this relatively larger amino acid peptide. We call it amyloid beta. So amyloid beta is quite sticky, and under some conditions, they can form large aggregates, okay, present in the brain, therefore can affect neuronal function. So the other protein I would like to talk about is called tau protein. And this slide shows, a neuron. We have dendrites, we can receive the information from other neurons, and this is the body. And the neuron has a very long axon, and this is the end part called axonal terminal can contact with other neurons. So the microtubule, and showed here, a very, very important structure in the axons, basically allows, some nutrient or neurotransmitters produced here and transmit down to the end part of that neuron. So tau protein is really important to stabilize the microtubule. So in the AD condition, all those proteins becomes problematic, okay? So loss of function, therefore, cannot stabilize this microtubule. As you can see, in the AD conditions, all those microtubules just broken. And the tau protein forms aggregates. So if you use some very specific method, you can see all those ghost tangles in the brain. And eventually that neuron just died. So another important point I would want to say is the amount of tangles parallels the duration and also severity of Alzheimer's disease is not the plaques. So what is the cause of AD? So in the past, the amyloid cascade hypothesis has been the leading one. However, this hypothesis has been challenged dramatically. So this publication in 2014 basically says, the amyloid hypothesis, time to move on. Amyloid is the downstream result, not cause of Alzheimer's disease. So the basic thing is, many industries, they want to develop some drug to treat Alzheimer's. They only focus on the amyloid cascade side, and no single clinical trial was successful. So this paper says, probably we need to think about a microvascular part because the brain, lots of neurons, we need to have a very good blood supply to maintain the normal cellular function. So later on, there was another hypothesis come out, and the two-hit vascular model of Alzheimer's disease dementia. So this idea was probably for Alzheimer's disease. Remember, for AD, 95% of Alzheimer's disease patients, they are old, okay? And we call the sporadic and late onset Alzheimer's disease. So aging should be the major risk factor. So they think cerebrovascular damage is the initial trigger, which is a hit one, then can affect the A-beta process and leads to the more production of amyloid. And remember, the cerebrovascular damage itself can affect neuronal function, okay, and also leads to the neurodegeneration. So personally, I quite like this idea. So why arginine metabolism? So arginine is a semi-essential amino acid present probably in every cell in our body. And this amino acid can be metabolized by an enzyme called the nitric oxide synthase, in the short term is NOS. And with the production of citrulline and NO. NO is a gas, okay? It's a gaseous molecule. So in 1998, NO was a star because 3 researchers around the world independently discovered nitric oxide, is the signaling molecule in the cardiovascular system. And all those 3 received a Nobel Prize in physiology or medicine in 1998. That was really, really exciting. So now we know nitric oxide is not just present in the cardiovascular system, it's also present in the brain. And the nitric oxide is very important for nervous system development. So if we talk about the isoform of NOS, there are 3 isoforms, 3 types. And N means, neuronal NOS. And E means endothelial cells in the blood vessel, okay, it's eNOS. And the eNOS-derived nitric oxide plays a very, very important role in terms of maintaining cerebral blood flow. We need to have normal blood flow in order to maintain our normal brain function. So NO is also involved in neurotransmitter release, learning, and memory. However, this gaseous molecule is kind of a double-edged sword. If it's too high, particularly NO produced by this iNOS can be neurotoxic and leads to neurodegeneration. On the other side, if NO derived from the eNOS, from the blood vessels too low, can affect cerebral blood flow, therefore we cannot function normally. So after I finished my PhD, there were a couple of papers published, and one is about nitric oxide hypothesis of aging. I thought, wow, this is very, very interesting. And the other paper published in 2001 is to say nitric oxide to Alzheimer's disease, the putative links between NO and dementia of Alzheimer's type. So the other day when I prepared my IPL, I thought, well, how did I know NO? And suddenly I remembered when Ewan and I worked in the lab and she mentioned nitric oxide. I thought really, really interesting. I searched for more, so I worked on this. So that was my plan initially. I thought, well, I probably could look at, compare the NOS changes between young and aged rats, try to look at how arginine metabolism changed in aged brain. And Yin told me, Payne, do you know arginine can be metabolized by another enzyme called arginase to form ornithine and urea? If you are going to do NOS, probably it's better to look at arginase as well, because NOS and arginase, they compete the same substrate. I thought that this is a good idea, okay? Yes, I did. So at that time, we were so good in terms of collecting small bits of tissue from different parts of the brain. I was really interested in the hippocampus, so we can collect the dorsal part versus the ventral part of the hippocampus, just because the dorsal versus the ventral hippocampus, they have a very different function in terms of spatial learning and memory. And we could also dissect different parts of the hippocampus, okay, CA1, CA3, and CA4, and the frontal cortex, and also the lateral side, interrhinal, perirhinal, whatever. So I published several papers that basically shows there are age-related changes in NOS and arginase in multiple brain regions, and some changes actually correlate with the behavioral performance. Later on, I discovered it's not that simple, arginine metabolism, because ornithine can be further metabolized to glutamate, GABA, and glutamine. And those two are the main neurotransmitter in the brain. One is to stimulate the neurons, and the other one try to inhibit neuron. And ornithine can also be metabolized to form a group of polyamines, putrescine, spermidine, and spermine. Polyamines, the physiological level of polyamines are extremely important for health. So in cancer patient, okay, polyamine levels extremely high, therefore make the cells to grow abnormally, becomes a tumor. If it's too low, it can affect cell growth. So how this relates to AD, and we know polyamines play a very, very important role in terms of make all those microtubules assembled, and also involved in the amyloid beta aggregation. And recently, there's research suggesting spermidine is a new longevity drug. If you treat aged rats with spermine, they are very healthy and they can perform much, much better than the control ones. And furthermore, spermine can block tau aggregation. Looks like polyamines somehow have a link with AD pathology or AD markers. Then I discovered more, okay? So arginine can be metabolized by another enzyme called arginine decarboxylase to form agmentin. Agmentin was really, really interesting. Itself can be can used to be as a precursor to form polyamines, and agmentin basically stands at the crossroad of arginine metabolism, try to regulate the NOS pathway and also the polyamine biosynthesis. Also, it's really, really interesting. And agmentin itself has been considered the putative neurotransmitter and also is involved in learning and memory. So from this diagram, you can see agmentin basically plays a very, very important role in terms of controlling nitric oxide production and also polyamines. And interestingly, agmentin has so many pharmacological effects, okay? For example, agmentin is a neuroprotective, anti-inflammation, and can enhance learning and memory, okay, and more. So for a while, and agmentin researchers thought agmentin is basically a magic shotgun because it has multiple molecular targets. So this diagram just illustrates agmentin biomedical research. So augmentin was discovered in 1910 by this famous Professor Kussel, and who was the Nobel Prize winner in that year. So he found augmentin in herring sperm. This is for the first time, okay, and we know augmentin. So during World War II, researchers found augmentin can be used to treat diabetes, okay, that's something interesting. It wasn't until 1994 there was a paper published in Science by Rees and also Sander Regnonsen. They demonstrated the presence of augmentin and also its biosynthesis enzyme ADC in the mammalian brain. However, the function, the endogenous augmentin in the brain is not clear. And later on, Jed Gillard published a number of papers showing augmentin is neuroprotective. And he even tried a small clinical trial to show augmentin can be used to treat neuropathic pain. I was really interested in augmentin. However, I didn't really have a way to measure endogenous augmentin. One day, I talked to my husband, I said, well, augmentin sounds fantastic. I want to measure augmentin. I have no way, because I don't really know any, analytic method. And he thought, okay, I probably can help you, okay? So he worked in pharmacy, and he knows how to do HPLC and LCMS. And he developed HPLC and LCMS method. So now we can measure arginine and its downstream metabolites, and which allow me to look at arginine metabolic profile changes in milligram tissue and also in biofluid, for example, blood and cerebrospinal fluid as well. So this really opens a big door for me. Thank you, And I know the augmentin is present in the hippocampus, and the hippocampus is so important for learning and memory. So this diagram really shows this is a synapse. It really means, the communication point between the neurons. And augmentin is present in the presynaptic terminal. So I thought, what's the role of endogenous augmentin in learning and memory? Is there any way I could figure this out? So if augmentin is involved in learning and memory, if you train animals to learn something, you would expect augmentin levels increased in the brain region involved in learning and memory, okay? Then I designed a study using the water maze. So this is a kind of swimming pool with a hidden platform located here. Then you can put rats to learn the place of this platform by using the external cues. So this is a spatial learning and memory. They have to use the cues to figure out where the platform is. So I have 3 groups, and the first one, water maze training group. You have to learn, where the platform is. And the second one, the swimming only, just swim. And the third one, do nothing, sitting in the box, right? Then, as usual, we collect samples from different parts of the brain. Then we quantify the augmentin levels. So this leads to a rapid publication. We found some brain regions shows increased level of augmentin. So this is probably easier to view, The numbers are too hard to see. So as you can see, there's no difference between the swimming-only group and the holding box group, which really indicates swimming did nothing for the augmentin. And it was water maze learning, the spatial learning leads to the increased level of augmentin in the hippocampus CE1, dentate gyrus, and vestibular nucleus, and also entorhinal cortex. So Associate Professor Beulah Leach, she's the expert in the electron microscopy, and we talked about and we collaborated. So we could use an antibody to bind to the augmentin and to look at all those black dots really means augmentin, in the synaptic terminals, then quantify those dots. And Bierke and also the student, we co-supervised Steve Seal, and found water-maze training induced increased augmentin level in the hippocampus CA1 synapses, which is very interesting. Further support the role of augmentin in learning and memory. So Madiha was the first PhD student I supervised with her. And she's very talented student and working hard. So we decided to do this in vivo microdialysis study. This is even more challenging. And we need to insert a probe, and we can collect fluid between the cells in the brain, then quantify the levels of augmentin before, during, and after the spatial learning in this water maze setting. Okay? So basically, we can look at the dynamic change of augmentin in the brain. So what she found, so this box really represents the basal level, and looks like swimming only didn't do anything. However, when the rats learning the platform, and you can see about 6-fold increase in augmentin in the hippocampus. And every time when you train, and you could see the augmentin levels going high, so really means augmentin does something. So we continue to train animals for a couple of days. Then you can test animals' memory to the platform locations. So this is a recall trial, and you can see the augmentin level increase again. So this study really provides the first evidence of endogenous augmentin directly participates in the process of learning and memory, which is fantastic. So my group, we had published a number of papers on the effects of aging on the brain, arginine metabolism in rats. I won't have time to talk about everything, just pick up a couple to show you. So this study, because augmentin is really, really interesting to me at that time. I said, wow, how aging affects augmentin in the brain? So again, this is a rapid publication. It's really simple. We have, some animals are pretty old, 24 months, and some rats are middle age, 12 months, and the other group, the young rats, only 4 months old. We collect the brain tissue, then we look at compared augmentin levels in the different region across all 3 groups. So it looks like aging affects augmentin level dramatically, okay? And if you look at the pattern, some brain regions shows, age-related reduction in augmentin. Other brain regions shows age-related increase, okay? What that mean? So later on, another PhD student, Neeraj, systematically investigated and how the arginine metabolism changes in multiple brain regions and across the 3 age groups. And he found aged rats, performed poorly in the water maze task. This is the path length, means aged rats generate a longer distance to find the platform, poor performance. So those aged rats also couldn't remember exactly the platform was, therefore had reduced the number of platform crossing during the memory test. And interestingly, Neeraj found some changes, of course, is region-specific. Not every region shows changes. The increased NOS activity, I just want to highlight prefrontal cortex, which is a PFC here, and also POR here. Then, lots of numbers, because we looked at arginine and the downstream metabolites and across all those 8 brain regions, lots of numbers. So those arrows basically increase, indicates high level in the aged rats, otherwise reduce the blue one, the green one. So another thing, quite interesting, you can see all the changes are consistent, okay, across all the brain regions. And augmentin in some regions shows reduced augmentin. For example, in the prefrontal cortex, the other one shows increased augmentin in the POR. So it took me a while to figure out what all those means. And the simple way, I want to look at how augmentin relates to the NOS, because augmentin can regulate NOS. So from this diagram, looks like lower, because it's a negative correlation. looks like lower level of augmentin, okay, and have higher level of NOS. And for POR region, this is a positive correlation, looks like high augmentin level and high NOS activity. So the explanation was the high level of augmentin may be a compensatory mechanism to control age-related elevation in NOS activity. If their region augmentin level is pretty low, probably cannot do this task. So then my research moved to the Alzheimer's disease, and which was supported by 2 HRC program. So the first one is a cognitive decline during aging and Alzheimer's, and we are focused on biomarkers and the therapeutic targets. And the second one, we're just about to finish by the end of the year. So as you can see, there are a number of researchers involved in this program. And the first one, we have Professor Robert Knight from psychology, and Nick Carterfield, and Professor Cliff Abraham. And the second one involves Cliff Warren-Tate from biochemistry, and also Associate Professor Joanna Williams and Stephanie Hugh. We have been working together for 10 years. So my work in the AD field, and basically we try to do multiple things. Post-mortem human brain tissue will tell us what's going on, what kind of changes of arginine metabolism in the human brains because of the real diseased conditions, okay? And we also have blood and also CSF samples from the human patient, and we can work on the biomarker side. So animal models are really good because those models allows you to look at each individual aspect, okay, try to understand the mechanism side. As you can see from this slides, we have all kinds of models. We can do the brain injection of those toxic peptides, and we also have a transgenic mouse model to target amyloid beta, tau protein, or eNOS. So another branch of my work is to explore the therapeutic potential of augmentin. So all those students contribute to the publications. We basically looked at all those aspects, okay? So I am going to show you the human tissue work, which is the collaboration with Professor Richard Fall's group in Auckland, and along with Morris, Henry, and Jocelyn. So this is the team from Otago University. So this is the first study to look at all 3 metabolic pathways, okay, and to look at how arginine metabolism changes in Alzheimer's disease. So the samples, the regions we selected was the superior frontal gyrus and hippocampus and the cerebellum. So at that time, we used the cerebellum as a control region because cerebellum is very special. For whatever reason, cerebellum doesn't have tangles. You can see the plaques, but no tangles at all. And superior frontal gyrus and hippocampus, of course, are affected severely in Alzheimer's disease. And we have 3 groups. We have a normal case at age 60, and a normal case at mean age 80. So there's a 20 years difference. So we can see age-related changes in arginine metabolism. And we also have a third group, which is Alzheimer's disease group, the mean age 80. Then we can compare those 2 to find out if there's any Alzheimer's disease-related changes. So Mike Fleet was the 4th year honor student at that time. So all the NOS and arginase enzyme assay and Western blotting work was done by Mike Fleet. So in general, we found reduced NOS, okay, and increased arginase. But if you look at all those, the bar graph closely, you can see AD and age-related disease. So just to remind you, there's a black bar means AD group, and there's a hatched bar in the middle is NC80, and this open bar, NC60. For example here, the hippocampus, the NOS activity is extremely low, and NC80 is low, and NC60 groups here. So age and also AD-related decrease. I am very, very interested in this result because it really shows the eNOS deficiency in the Alzheimer's disease patient. As you can see, the eNOS in the superior frontal gyrus and hippocampus, the protein level was extremely low. And the eNOS-derived nitric oxide is so important in maintaining normal cerebral blood flow. Okay, this is very interesting. So in terms of arginase, and we found AD-related increase because there's no difference between NC60 and NC80. There's only increase in the AD group relative to the other two control groups. So we found reduced NOS, but L-citrulline is the product of this enzyme, showed no change. We found increased arginase activity or protein. However, the product L-ornithine was reduced very specifically in the AD cases, okay? And regarding augmentin, we found age-related decrease in augmentin. And for putrescine, AD or age-related. So I tried to put something together and find meaning of this. And when I put something like this, the NOS is down, and no change in L-citrulline. Arginase is up and reduced ornithine. Okay? Took me a while to figure out what all those numbers means. And eventually, I discovered another pathway which involves ornithine transcarbamylase. This is the key enzyme involved in the urea cycle. So the urea cycle is the important, very important cycle present in the liver, aiming to remove this toxic ammonia through this pathway to produce non-toxic urea. This is a very important pathway. However, in the AD brain, ammonia level is very, very high. Therefore, it is possible under the condition of AD, because ammonia level is quite high, so this pathway somehow is induced as a protective mechanism, okay? So my PhD student, Jody, has been working on the urea cycle, and he has some evidence suggesting OTC is indeed induced, okay, in the Alzheimer's disease brain. So there are a number of questions for me to answer. And first one, whether all those arginine metabolic profile changes and the urea cycle induction are very specific to AD. If not, whether all those changes could be a marker of neurodegeneration. We are still working on this. So as I mentioned earlier, the animal model provides a very good tool for us to tease out each part, okay, of the pathological changes in the AD brain. So the PhD student Pranav worked on the PS19 tau mice, and which is mainly focused on the tau protein, okay? And he found altered arginine metabolism. And this is a collaboration with Professor Daniel Lee in States. And he also worked on APPPS1 mice, and this mice to model amyloid beta side, okay? And PhD student Ashwini and worked on eNOS deficient mice because we could see dramatic reduction in eNOS protein in the AD brain. So they found altered brain arginine metabolism, however, in a different way. So today I won't have time to explain the data, but anyways, lots of changes, okay, in all those models. I'm really, really interested in eNOS side because eNOS-derived NO is so important for cerebral blood flow. So I thought we probably could make a new model, all right? So APPS1 presents amyloid beta pathology. So how about we add a cerebrovascular component to this model by crossing APPS1 mice with eNOS knockout mice? So this work, PhD student Sarah Ahmed and Hannah also contributed during her honors year. So this is an example of amyloid plaques in the brain. Okay, as you can see, this is APPPS1 mice, looks like this, and this is triple one APPPS1 eNOS partial knockout mice. Definitely have more plaques in the brain, which is quite interesting. And interestingly, this new model had more severe memory deficit when we tested, okay, animals in the Morris-Water maze. So this is a very interesting model because now funding is really tight. You don't really want to keep these animals until 12 months of age, then use it. So those animals shows quite severe A-beta load increase and also behavioral impairment. So we are also interested in the biomarker side. This is a PhD student, David Bergen's work, and he reported altered plasma arginine metabolome precedes the behavioral and brain arginine metabolic profile change in the APPS1 mice. So he used this APPS1 mice and tested two age groups. One, the younger group at 7 months, the other one at 13 months. So for the younger group, you could only see very scattered amyloid plaques in the brain, okay? And for those animals at 13 months old, you could see lots of plaques in the brain. And the older one also shows the behavioral impairment as well. So they found lots of some neurochemical changes in the brain in the 13 months old, but only very, very mild change in the 7. But interestingly, he also looked at the plasma profile, and he found lots of changes in plasma in the younger group. For example, the putrescine and the spermine, and those ones are the polyamines. When we did this very special analysis, walk analysis, and plasma putrescine and spermine, basically were able to separate APPS1 mice from the control, okay, which is something quite interesting. So I would like to further explore the potential, the biomarker side using the arginine metabolism. So at Alzheimer's conference, I met Dr. Albert and also Gerard at a conference. So they were from Spain. We basically look at the same poster and talk to each other. And said, oh, you are interested in biomarkers? I said, yes, what did you do? Okay, turns out they are very, very interested in my arginine metabolism work and provide the samples, both plasma and CSF samples from the same subject. And this time we looked at Alzheimer's disease patients with early onset. So we found a number of arginine metabolite level changes in the plasma and CSF. Just give you one example. We haven't published the results yet. So you can see this arginine metabolite 1, the levels reduced in the plasma and also CSF basically shows a parallel reduction, which is quite interesting. Then we found that the lower level of this arginine metabolites and correlate with high level of tau, either total tau or phosphorylated tau. Tau is not good, okay? And more interestingly, when they provide cognitive function test results, which is MMSE, mini mental state examination results. So lower level of this particular arginine metabolite correlates with the poor performance, okay? Poor function of cognitive. So I would like to further explore the potential of arginine metabolism as a marker for AD and looking for more funding to support this line of work. Augmentin is my favorite. But sadly, this field is quite small. Not many people working in this field. And many years ago, I remember when I presented my work at a conference, many people look at me, augmentin, what is augmentin? I've never heard of augmentin. No idea what you are talking about, okay? So collectively, I think this is a group of 16 researchers around the world, and we decided to write a review article. The title is Augmentin, Clinical Applications After 100 Years in Translation. My PhD student, Madeha, she's really, really interested in augmentin, and she carried out a study to look at anti-aging effects of augmentin. She had 3 groups: the young animals treated with saline, aged rats treated with saline, and also aged rats treated with augmentin. She tested animals in the object recognition task, okay? So first of all, she just put the animal in the box and get animal to familiar with all those objects. And one day, she put a novel object to replace one of those familiar ones. And you could see aged rats with saline treatment performed poorly relative to control. However, aged rats with augmentin treatment performed as well as control. Then she did another test, simply all those familiar objects, and she simply swapped the position and displaced object. So again, you could see aged rats treated with saline performed poorly relative to the young one, but augmentin treated ones performed much, much better, as good as the young one. She also looked at some neurochemical changes in the brain, and this time, and you could see the aged rats with saline treatment had higher level of NOS activity, and augmentin treatment basically can normalize this kind of age-related changes in NOS activity. And interestingly, the eNOS protein is reduced in the aged brain. However, augmentin treatment can restore the eNOS protein back to the normal level. So this is very interesting. So David Bergen looked at another model, and this time he used this toxic amyloid beta peptide, 25 to 35. And this 11 peptide and basically is the core part of the foldings of amyloid beta. So he infused both into the brain and also tested in the behavioral test. So this is radioamylase task. So A-beta, the toxic peptide group with saline treatment, made more errors in this task. However, augmentin treated one can reduce the errors and perform as good as the control. So this is the same task, novel and displaced object, and you can see the augmentin treatment can protect against amyloid-induced memory impairment in the rat. So Sarah Ahmed, still working on her PhD, tried to look at therapeutic potential of augmentin for AD. The project is still ongoing. So augmentin, I think this augmentin research has gained great interest over the last couple of years. So this is a very important paper published in the very high impact factor journal Cell, indicates augmentin has a role in longevity. So metformin is a drug to treat diabetes, and metformin has a longevity effect. So this study basically shows augmentin mediates the longevity effect of metformin, because when you take metformin can stimulate the gut bacteria to produce augmentin. Then augmentin can increase lipid metabolism, therefore to increase the lifespan. This is a very interesting study. So we have a project ongoing to look at the role of augmentin in maintaining healthy brain aging with the support of Brain Health Research Center, because the rats are very, very expensive. And JEDGLAD kindly gifted 2 kg augmentin to support our work. So we basically treat animals from middle age for 9 or 10 months, then tested, how those aged animals behave in the tasks, and also cerebral blood flow and other factors as well. So I have 2 honor students working on the project to look at the mechanism side. Okay, I have talked about Alzheimer's disease. So AD is basically the main form under the big umbrella of dementia. At this stage, we have no cure for Alzheimer's disease. What could we do? Stop Alzheimer's probably before it starts. So now, AD researchers have waved the big flag, okay, encourage people to do whatever you can to promote successful aging. Have a healthy, lifestyle and exercise regularly, all right? Do whatever you can. So this is what I have been doing. Okay, we have a very nice group, dancing friends, and we meet every week, and sometimes we perform, okay? And my favorite vegetable garden, I worked a lot. In my vegetable garden, I walked a lot, try to exercise. I set up a limit for myself. I need to walk 10,000 steps every day. So this is my goal, successful aging. I hope this is also your goal, okay? We do need to age successfully. That's the end of my talk. Thank you very much.
Tēnā koutou katoa, ko Lisa Madison-Smith taku ingoa, no Hawaii hau, no o te poti takakainga i nei ane, ko tumuaki o te tari kiko ahau. Nō reira, tata katoa e huihui mai nei, tēnā koutou, tēnā tata katoa. My name is Lisa Madison-Smith, and I am the head of Department of Anatomy, and it is my pleasure to be able to speak at these IPLs and to celebrate the truly impressive career and research career of our colleagues. So as a head of department, you talk with people when they're applying for their promotion, so you get to see the amazing CVs, because we know our colleagues, but we don't know our colleagues that well in a large department. But when you sit down with somebody who's applying for promotion, you really get a chance to look at their CV, and it is truly an honor, and as always, but certainly in the case of Ping as well. But at the IPL, at the lectures, you get to see another side to our colleagues, and you can see how they got to where they are, and you start to see patterns, I guess, in their behaviors and the way that they do things. And again, I think we've seen this with Ping, and we see certain patterns like her dedication to family, her commitment and the support that she has and that she gives to her family. And similarly to her students, we see these patterns of extended collaborations, extended over time, and extended across the globe. And certainly that's been demonstrated in Ping's IPL tonight. I also often say, watching these IPLs, there's something in it for everybody, and you always kind of see something that really connects to you. And sadly, in Ping's case, the thing that really got me was, learn slowly, forget easily. That's me. I get up here and I completely forget everything that I was going to say. But I do remember talking with Ping, I think it must have been just after lockdown, perhaps just before, but she was so excited. She said to me, "Lisa, I've got this huge amount, 2 kilos of agmatine. I'm so excited." And I kind of smiled and nodded and said, it was clear that this was quite a score. So after tonight's lecture, I have to say that now it's my favorite too, and I got to get some of that magic of agmatine and try to deal with my hippocampal issues. So I thank you certainly for educating me on this important research, and in all seriousness, thank you for the work that you've done addressing these very serious issues that so many New Zealanders are facing. So please join me in thanking Ping for this wonderful IPL, and please join me now. And Ping. I also get to present you with a small memento and congratulation and recognition of this wonderful achievement. So congratulations.
Kia ora, tēnā tātou katoa. My name's Paul Brunton and I'm the PVC for Health Sciences. And I have two duties tonight. The first duty is to thank you, our audience, for coming to this very enjoyable, I'm sure you'll all agree, inaugural professorial lecture. So firstly, thank you for your coming. And secondly, my final duty is to invite you to come and join us for refreshments, to continue to celebrate Ping Lu's success and join us in fellowship over some refreshments. But before we do that, I think we should thank Ping Lu one more time in the usual way. Thank you.